Okay, so this is going to look like a terrifying, set of images here, but you understand all of this if you've been following all these video lectures up until now and reading your textbook and going over notes, right? So this is what we refer to as receptor regulation, right? So this is a postsynaptic cell. This is my attempt to draw it. It's a box, right? This is the postsynaptic membrane with all these receptors that are found here along this postsynaptic membrane. So that means there's a presynaptic membrane up here somewhere and neurotransmitter that will be released upon the arrival of an action potential, right? It'll be coupled to the arrival of the action potential. So this is supposed to represent, this is the nucleus, right, of this postsynaptic cell, right, where the DNA is located, where genes are transcribed and then translated, right, into proteins, and these proteins include receptors, and some of these receptors that are going to be, inserted into the membrane at the axon, or sorry, at the postsynaptic membrane. So this is an example of a lot of drug being introduced into this particular synapse. This is an agonist drug. So remember, an agonist drug is going to bind to all of these receptors, G protein coupled receptors, ionotropic receptors, right? That it actually has affinity for. And it's going to provoke the release of the G proteins. It's going to provoke, the opening of the ionotropic receptors. Let's say we get a lot of this agonist drug present in this synapse and a lot of G proteins released. Well, these G proteins, of course, are going to activate, more second messengers, right? And then more third messengers, and then more fourth messengers. They're going to actually set off this biochemical cascade, right? A lot of changes are going to occur in this postsynaptic cell with exposure to this agonist drug, right? And then these second messengers are going to, ultimately you're going to get some chemicals that are going to enter the nucleus, bind to the genetic material, open certain sequences for transcription, close others. You're going to get changes in gene expression, right? And receptor protein synthesis. And so the result I'm showing you here is what we refer to as downregulation. This means fewer receptors than you had initially. So initially you had, let's say, this set of receptors, right? And after a lot of agonist exposure and a lot of G proteins released and a lot of communication with the nucleus, the cell responds by reducing the number of receptors expressed in the membrane. So there are going to be fewer receptors and perhaps different receptors that are put up here. This is an example of actually learning, right? This particular synaptic connection here is changing in response to the introduction of this drug. Before it had this level of response, had a certain number of receptors. Now the response has been downregulated. There are fewer postsynaptic receptors that can respond to the drug.